last speaker before the intermission is Professor Iwasak of the School of Medicine. She studies immunology and the immune system that detects viruses and assembles an army of cells and molecules to fight infection. She came to Yale in 2000 and started to study how viruses are detected and how they use that information to protect against the host, against the virus, and ultimately learn from the natural immune responses to develop better vaccines. Welcome.
It's a true honor for me to be standing here and to be able to tell you some of the things that we do. So whether you like it or not, we have an intimate relationship with viruses. Just to give you an example, there are 10 to the 31 viruses that inhabit Earth alone, which means this translates to 100 million light years worth of virus if you put them end to end. That's pretty remarkable considering that each virus is about 100 nanometers in diameter. And these viruses can not only infect humans, but virtually every living thing on Earth. Bacterium, animal, plants, they're all susceptible to virus infections. And not only are they susceptible, but they're likely colonized right now. Everyone sitting in this audience probably has millions of viruses that you're cultivating without knowing. And viruses actually come in many different flavors. Some viruses have RNA as genome, and others have DNA. But what's common about viruses is that they have genetic material that's either encoded by RNA or DNA, which is encapsulated in a capsid. And this capsid protects the virus from becoming degraded. And also, some viruses take the host membrane with them. So they're called envelope viruses. And they have very different forms, as you can see. Influenza virus, for example, have 8 segments of RNA. The adenovirus has a DNA in its genome, but they all can infect different types of cells and different hosts. So this is a staggering fact that I'm sure not everyone knows in this room, that we are more viruses than humans, genetically speaking. So if you look at our genome, there's about 1.5% of our genome is dedicated to coding human genes, whereas the vast majority, about 50% of our genome, consists of viral sequences that have integrated into our genome over millions of years. Okay, so really, literally, we are more viruses than humans. And some of these viruses are still alive, and they're transposing, as I speak right now, within your cells, and mucking up the genome. In other viruses, we have actually learned to live together and even co-opted their function for our own purposes. So as an immunologist, I'm very interested in understanding how viruses are detected by the immune system. And this question is, when applied to other kinds of microbes, like bacteria or fungi or parasites, it's relatively easy to understand because these bacterium, for example, have cell walls that the mammalian cells have never seen before. These are very unique structures that only bacteria has, and also that tail that spins around for them to allow them to swim around in the body, called flagellum, is also never found in mammalian cells. So we can use those unique sort of components of bacteria as a flag to detect these kinds of microbes. On the other hand, how the immune system detects viruses has remained a conundrum for a long time because, by definition, all viruses can only replicate within the host cell. So essentially, everything that they have, their protein, their lipids, their nucleic acids, it's part of us. They took it from us. So there's nothing really unique about viruses at all. And so how do we deal with this situation? We have to be able to detect them in order to mount immune response and clear them. So we've been studying this question for a while, and we came up with an answer that we use mislocation to detect viruses. So to give you an example, and I'm really glad that I'm not representing the School of Arts, this is a cell. And obviously, this is the nucleus. And in the nucleus, you can find DNA. Now, in every healthy living eukaryotic cell, the DNA is contained within the nuclear envelope. So it's really not anywhere else. And if you look at any tissue, you take liver or lung or skin, what you find are cells that look like this, not exactly, but like this, where the nucleus contains the DNA, and they're very quiescent. Now comes along a virus, which includes its own DNA, and it can inject that DNA in order to replicate themselves. So what happens in this situation is that what we've realized is that the host cell has strategically placed sensors that can detect DNA that's in a wrong place. In this case, it's the cytosol, but it can also be in vacuoles or endosomes or other places in which DNA should never be found. And so this is a really location-based detection mechanism that we as vertebrates have evolved to use. And these sensors, once they detect the strange location of DNA, can send alarms, and these are known as interferons. And these interferons can bind to the neighboring cells, and so the neighboring cells can put up shields against the virus, such that the virus can no longer infect these neighboring cells. And the cells that were originally infected, the one that doesn't look so healthy, have died because of virus infection. So it's a very altruistic way of protecting the whole organism by dying, but then alarming the neighboring cells about this presence of the virus. We've also discovered a few years ago, Ellen Foxman in my laboratory, that even cooling the temperature by a slight 4 degrees from the core body temperature of 37 degrees versus what you find in the nasal cavity, which is about 33 degrees, can dampen this alarm system altogether. So, and in the winter month, of course, when you're inhaling very cold air, the nasal temperature even goes further down. And this allows viruses, like the common cold virus, to replicate in the nose. And this may even explain the fact that we catch the common cold usually during the winter months and not in the summertime. So what I've been talking about thus far is innate immune defense. So innate immune defense is actually contained within every cell of our body. It's innate because it's not learned. It's basically detecting any foreign invaders. For those of who are Game of Thrones fans, you know that these frontline soldiers will basically attack any foreign bodies that come across its way. So it's a very frontline defense. It's not that intelligent. It doesn't really know what you're killing, but you're supposed to kill the invaders. So this is very important. It's a first line of defense. It alarms the cells and neighboring cells about the infectious agents. However, we have another arm of the immune system called the adaptive immunity. And this, unlike the innate immune system, this is a very intelligent immune system in that it learns to detect specific viruses. So for instance, influenza virus may be different from herpes virus. And these adaptive immune response can only detect specific selective antigens from those different types of viruses and kill. And they can also seek and destroy. So not only they are just there waiting for them to become infected, but they can circulate throughout the bloodstream and detect these infected cells and kill them. And this is the basis of all vaccines. So I want to end my talk by giving a big plug to vaccines. Vaccines are highly effective. We forget the usefulness of vaccines because we are so well protected. We forget the deaths and diseases and morbidity that are caused by all these common infectious agents because we're sort of beneficiary of all these vaccines. And you don't need to be able to read the exact signs indicating which pathogens, whatnot. But essentially, before the vaccine, the world looked like this. There are millions of people who were infected with these diseases, and often they are lethal or crippling. Whereas right now, we are at this stage on the left, where there are some viruses and bacteria that still harm the human beings, but you can see the magnitude that is hugely reduced by vaccines. So I think it's important to consider this fact in the era of alternative facts, that it's really important to keep in mind. And this is really, data really speak for itself. So I want to end by thanking the people who actually inspire me every day. These are the members of my laboratory, past and present, who really give me reason to come to work and sort of enjoy this amazing thing about the immune system and the viruses. And highlighted in green are those that actually went on to start their own laboratories. And I hope this infectious love for science will spread throughout the world. Thank you very much.